In order to develop a national biodiversity assessment, we need to understand and map the pressures on our biodiversity. Maritime activities are defined as pressures on marine biodiversity in South Africa. Some pressures are easier to map than others, and some pressures exert greater impact on biodiversity than others, leading to varying degrees of modification. The type, extent, and intensity of the pressure are therefore integral to understanding and sometimes being able to predict the resulting modifications for biodiversity. In South Africa, we recognize there are many pressures on biodiversity. In the last NBA, we recognized 31 pressures in the assessment and collated data, including spatial data, from different sources on each one in order to develop maps. In order to carry out the assessment, we needed to score the impacts of our pressures. This is done to operationalize what we know about a specific pressure and the associated impacts on biodiversity. We understand that ecosystem types respond differently to different types of pressures, and therefore these differences need to be captured through pressure scoring. In South Africa, we use the Tech et al. 2010 methodology, which uses guided group consensus to score for functional impact and recovery potential. Functional impact is defined as the degree to which the natural state of any given ecosystem type is impacted or modified by a specific activity, whereas recovery potential speaks to the time required for the ecosystem type to recover to a natural or near natural state following the cessation of that activity or pressure. These scores are combined and used to develop an ecosystem pressure matrix, where each pressure impact is evaluated against each ecosystem type. Using the raw data received from partners and data contributors, we are able to map the spatial extent of pressures. This allows us to identify the underlying ecosystem types that each of these pressures may be affecting. Some pressures can have high impact with only one application, while others have higher impacts under chronic application. This means that we expect negative impacts from those pressures to accumulate over time. Understanding pressure intensity is therefore a key component in understanding when modification to biodiversity occurs, and mapping intensity provides insight into where the highest impacts from pressures can be expected. Mapping of pressure extent and intensity is done separately from pressure scoring, but the ecosystem pressure matrix is a key component to developing maps of pressure impact using intensity combined with the scores from the ecosystem pressure matrix. We use a cumulative impact map in our assessment. This is developed using the Halpern et al. 2008 formula to derive the cumulative impact score. This combines the previously discussed elements, these being the marine map of ecosystem types, the maps of pressure intensity, and the ecosystem pressure matrix to develop cumulative impact scores that can be spatially mapped. The cumulative impact map provides the necessary spatial data to determine the ecological condition of an ecosystem type. Areas with high levels of cumulative impact are expected to have high levels of biodiversity modification. And these levels of modification are divided into 4 condition categories. Where we have no to very little pressure, we expect the ecosystem type to be in a natural or near natural condition. Under moderate pressure, we expect it to be moderately modified. Similarly, where there is severe pressure, we expect it to be severely modified. And where we have our highest levels of pressure, we expect the underlying ecosystems to be very severely modified. The map of ecosystem condition underpins the assessment for the headline indicator of ecosystem threat status. The threat status of ecosystem types is assessed using guidance from the IUCN Red List of Ecosystems. The map of ecosystem condition facilitates our ability to assess the degree to which an ecosystem type has been modified and what proportion of its spatial extent is in a specific condition category. Using the IUCN guidelines, we can determine which ecosystem types are considered least concern, vulnerable, endangered, or critically endangered. The NBA also included a near threatened category to identify those ecosystem types that are close to the threshold of the vulnerable category. Ecosystem protection level is determined by the proportion of an ecosystem type present in South Africa's network of marine protected areas. An ecosystem protection level is our second high-level indicator. The map of marine protected areas is therefore overlaid onto the map of ecosystem types and used to determine which ecosystem types fall into the well-protected, moderately protected, poorly protected, or not protected categories for protection level. These are, however, evaluated against the biodiversity target. This is the minimum proportion of each ecosystem type that needs to be kept in a natural or near natural state, and therefore only ecosystem types that fall within this condition category can be considered under the well-protected protection level category.
To give a little bit more insight into ecosystem protection level categories, I'm drawing from a really nice schematic that Sizo Sobanda, one of our emerging researchers, contributed to the National Biodiversity Assessment. Basically, if less than 0.2% of an ecosystem type is within our marine protected area network, that ecosystem type is considered not protected. This is just to prevent a sliver erroneously contributing to protection. If more than 0.2%, but less than 10% of an ecosystem type falls within the marine protected area network, that ecosystem type is considered poorly protected. Then, if between 10 but less than 20% of an ecosystem type falls within the marine protected area network, that's considered moderately protected. And well-protected ecosystem types are those where more than 20% of an ecosystem type is protected within the marine protected area network, and all of that is in good ecosystem condition. So we're basically applying a filter here that if an ecosystem type meets its target, but that ecosystem type is in poor condition because of activities within the marine protected area, for example, then that protection level is downgraded to moderately protected. And here you can see the value of such a headline indicator in the complexities of integrated ecosystem assessment, because you can see how this indicator reflected South Africa's progress between assessments in establishing marine protected areas. In 2018, South Africa had 25 marine protected areas, mostly coastal, but also the Prince Edward Islands Marine Protected Area in our Southern Ocean Territory. But in 2019, a new and expanded Marine Protected Area network was proclaimed, bringing our total to 42 marine protected areas. And you can see how the gray bars in these charts decreased. The not protected decreased, and the green bars, the protection, increased. And this is reflected even more strongly on the map, showing that substantial increase in marine ecosystem protection levels, with overall coverage of South Africa's mainland ocean territory advancing from less than 0.5% to 5.4%, and really reflecting that progress in ecosystem protection. Thank you.
Here I'm going to briefly talk about how we are trying to advance the current condition assessments from the National Biodiversity Assessment, and how we can use these assessments to ground truth the current condition maps and develop an indicator assessment framework. The current methods applied in the NBA are hugely valuable to determine ecosystem threat status using multiple pressures that have been measured and mapped throughout South Africa. However, These ecosystem assessments are a proxy for ecosystem condition based on the assumption that where you have a high intensity of pressures, we assume that the ecosystems are in poor condition and vice versa. While these maps are important to estimate condition at a national scale, we still don't know for sure what the real ecosystem status is, particularly at a much finer scale. What is really going on underwater? We need observational, in situ, and model data from our oceans to measure and map ecosystem condition and to better understand where we have healthy functioning ecosystems or where they are becoming degraded. However, we are still trying to figure out how to measure ecosystem condition. What indicators can we use? And we need to use these indicators to determine what it actually means to be in good ecological condition. Here is an example of some of the indicators and metrics that can be used to measure ecosystem condition. Some of the research questions that we hope to achieve include: how do we measure ecosystem condition, the identification of suitable indicators, what does it mean to be in good ecological condition, defining reference conditions, what is the status or condition of our ecosystems, and how do condition assessments using ecological data compare with those estimated from the MBA. To do this, we have started to develop an indicator selection and assessment framework where, using a specific ecosystem type, we need to select indicators based on a set of criteria to measure condition. We then need to test and validate these indicators and then use these indicators to define reference conditions, thresholds, and targets for future condition assessments. And this is what we set out to do. Using subtropical rocky reef ecosystems as our target ecosystem type, we collected in situ ecological data using remote imagery techniques, specifically baited remote underwater stereo video systems. We collected fish and benthic community data from selected areas on the east coast of South Africa. Data were collected from inside and outside current marine protected areas to obtain a gradient of human pressures. We wanted to identify indicators that respond to these pressures and that can be used to measure condition. We then use these indicators to determine what constitutes good and poor ecological condition. And these data were combined into multimetric condition indices to compare with those from the National Biodiversity Assessment. From the results of this research, we were able to identify which indicators were good at detecting impacts from pressures. Some of these included biomass of fish, proportion of large and mature fish, and trophic level. Comparing the response of these indicators between protected and impacted areas, we were also able to identify attributes of good and poor condition as a reference for future assessments. For example, rocky reefs that are in good ecological condition tend to have a higher overall biomass, average length, proportion of adult fish, and a lower proportion of turf algae and more upright, large, and branching morphologies from the benthic community, compared with impacted sites that are in poor condition. By combining this information into biological condition indices, we are then able to determine the relationship between condition indices based on in situ ecological data with those based on pressure data calculated in the MBA. This serves to validate the methods from the MBA to determine how suitable those assessments are as a proxy for ecosystem condition. We are able to ground truth the condition scores by comparing the two approaches, and we can then figure out where and why the condition assessments differ to refine these methods. So this has been done for one study using only one ecosystem type so far. So there is a need to further ground truth the MBA pressure-based assessments by applying these methods to other ecosystem types and in other areas around South Africa. We need to continue to test and validate the use of various indicators, especially across biogeographic zones. We are currently relying on the use of pressure data to conduct more of a risk assessment of ecosystem threat status, and there is a need to measure ecosystem condition, learning from international examples such as the Ocean Health Index and assessments conducted in the EU for the Marine Strategy Framework Directive. We need to include other ecosystem components such as bacteria, plankton, marine reptiles, mammals, and seabirds. This will advance our understanding of the impacts of pressures on ecosystem structure and function, but to do this, we still need to identify key cross-cutting indicators that can be used to inform reporting objectives and to conduct integrated ecosystem assessments.